Hi everyone, welcome back to another video. So today's lesson is going to be on cell communication. This is pretty much the next to last topic in cell bio, and it's just going to be about how cells communicate with each other and kind of the implications that has and just like the entire process. So yeah, let's get started. So first, let's talk about sensing between organisms. So obviously, if these organisms are unicellular, then this is basically the same as, cell communication, right? So let's talk about yeast, Saccharomyces cerevisiae. So in yeast, you've got two mating types, an A type and an alpha type. And the type A cells secrete an A factor, and the types alpha cells secrete an alpha factor. And basically, those A factors bind to a specific receptor protein on the surface of an alpha cell and vice versa. And what happens there is the alpha cell and the A cell, they fuse and they mate. So you can see it in a little bit more detail in this picture right here. But basically, this is kind of a use of, sensing between two organisms so they can communicate to each other and end up doing something, in which case, in this case, is mating. And another example is in bacteria. So there's this thing called quorum sensing. It's when you use the concentration of signaling molecules to coordinate density and behavior, and this happens in bacteria. So this is very popular among bacteria when they're creating biofilms, which are basically layers of cells that are going to be across some surface. So plaque on teeth is an example of a biofilm, as is sometimes, on like logs or something, you might have like another biofilm as well. But basically, biofilms make it really easy for bacteria to do something. And in order to make sure that they're not getting too crowded and to kind of coordinate their behavior, they do use quorum sensing. Another place where quorum sensing is used is when bacteria secrete toxins. So yeah, like, for example, like cholera. So cholera has to I believe cholera also does form a biofilm in your intestines, but basically it uses quorum sensing to kind of coordinate its behavior to secrete toxins, and that's what leads to the symptoms of cholera. Okay, so let's talk about local and long distance signaling. So now we're talking about eukaryotic cells. So eukaryotic cells, they may use direct contact to kind of send messages to each other. So this can be cell junctions, like, think gap junctions, think plasmodesmata, right? And they can also communicate via cell surface molecules, so still direct contact, right? And this is something that's important in like immune cells and stuff. It's also autocrine signaling, which is basically when a cell secretes some, signaling molecules that come back to itself and then, kind of inspire some sort of response within the cell that just produced those signaling molecules. So you see this with plants. Plants make this hormone called ethylene, and it is a gas, and basically it usually goes back to the cell that makes it, and it tells the plant to ripen. So yeah, we also see paracrine signaling, and this is where the signaling cell secretes molecules that travel really short distances. You see this a lot with growth factors, which are really important during, say, like embryonic development. They kind of tell things to grow, and usually growth is kind of coordinated regionally, so that's where paracrine signaling is very useful. And then we also see synaptic signaling. This is basically in the special case of neurotransmitters crossing synapses, which are kind of like the little gaps between different neurons, which are brain cells. So you can see a picture right here. Basically, you've got a part of a dendrite, which are those kind of extensions off of what looks like the head of a neuron, and then it's reaching a target cell, and they're not quite touching, they're almost touching, but basically you're going to see neurotransmitters diffuse across this tiny little gap and then make it to the next cell. So this is what we call synaptic signaling. And then finally, endocrine signaling. This is going to be over the longest distance, and it utilizes the bloodstream. So obviously, here is a picture, and epinephrine or adrenaline, they're the same thing. That is a really good example of endocrine signaling. And something to note with endocrine signaling is that it causes relatively slow changes. And in general, all of these things do not cause like super, super fast changes. So yeah. All right, so let's look at the stages of signal transduction. And signal transduction is basically the process by which, a cell takes in a signal, kind of translates it into a way it can understand, and ultimately that becomes a response. So we can see here the first step is reception, which is detecting that signaling molecule, usually when it binds to a cell receptor protein. And then the second step is transduction. This one tends to be the most involved with the most steps, but that's basically when the cell receptor changes shape, and then that might lead to a cascade that leads to a cellular response. And this may involve multiple steps. And then lastly, we have the response, and the response can be a couple of things. It can be like, for example, enzyme catalysis, rearranging the cytoskeleton, or even activating or deactivating certain genes in the nucleus. So basically, all of those responses would have to come through kind of one of these signal transduction pathways, and we'll talk about each of these steps more in depth. So first, let's talk about reception. So what happens during reception is the signaling molecule, it matches the shape of the receptor's binding site, and then because it binds to that receptor, we call it a ligand. So when that ligand binds, it usually causes the receptor to change shape, and then this is where all the changes start happening. So binding is usually going to activate the receptor, and sometimes it can even cause 2+ receptors to aggregate together, right? So that's something that'll happen, but basically big takeaway is ligand binding activates receptor. And something to note is that most receptors are going to be on the plasma membrane because ligands are usually big and water soluble, meaning that it's very hard for them to pass through the plasma membrane. There are exceptions, and we'll get into them a little bit later. All right, so let's talk about some kind of popular plasma membrane receptors. So the first one is the GPCR family. And so basically what is distinctive about the kind of GPCR system is that you've got this G protein that binds to GTP. So GTP is basically like ATP, but instead of an adenine, it uses a guanine. So, yep, and then you've got a GPCR, which is the actual receptor. GPCR, by the way, stands for G protein coupled receptor, and then it looks something like this. So something to note is that it's got 7 transmembrane alpha helices. And if you remember from like the amino acids, like kind of macromolecules video, most of these alpha helices, the amino acids are not going to be proline or glycine, and the vast majority of them are going to be nonpolar since they are in the membrane. So yep, this is kind of what they look like, and then the segment that interacts with G proteins is on the inside, and the place where the signaling molecules bind that place is on the outside. And GPCRs, they tend to do a lot on like yeast mating factors, epinephrine, neurotransmitters. Those are all places where you're going to see the use of GPCRs. And yeah, they've evolved very early on. They're involved in many diseases. There's some crazy statistic, but like some really big portion of medicines are basically targeted at GPCRs. So let's look at how GPCRs specifically work. So what happens first is the G protein is kind of attached loosely to the membrane. It's not associated with anything, and it's usually got like a GDP bound to it. But whether or not it has a GDP or a GTP bound kind of determines whether or not it's active. So if it's just got a GDP, then it's inactive. And let's just go with that for our example. So the second step is our signaling molecule binds to our receptor, and that activates our receptor and makes it change shape. And then on the cytoplasmic side, it's going to bind to that G protein, and what happens there is the GDP gets swapped out for a GTP, and all of a sudden the G protein is also activated. Now the activated G protein goes and binds to an enzyme and activates that enzyme. And then that enzyme can lead to signal transduction, which leads to a cellular response, right? And then this is something that's really powerful because this G protein oftentimes can go activate multiple enzymes, right? So we can amplify this response right here. And also, just pretty much as long as the GTP is still bound to this G protein, it's still active. So our body, organisms, we need a way to kind of shut that down. And the nice thing is that it's kind of built in. The G proteins can hydrolyze their own GTPs. So they can turn their GTPs into GDPs once they realize, that they've kind of done enough. So that's what they do. And then you kind of reset to the beginning state. So that's like a really nice turn-off switch that's kind of built into this GPCR mechanism. All right, so now let's talk about a different type of plasma membrane receptor. So these are called receptor tyrosine kinases, or RTKs, and they look kind of like this. So I like to remember what they are, kind of what they look like, because receptor tyrosine kinases, like I think of the T in the middle. This looks like sort of like a T to me. So yeah. But you'll see that each one of these words actually makes a lot of sense in the context of what an RTK is. So a kinase is an enzyme that catalyzes the transfer of phosphate groups. And this is usually involved with ATP, right? Because ATP carries those phosphates. In the bonds between the phosphates is where we get that energy, right? So our RTKs, they have the kinases sticking out into the cytoplasmic side. So it's this area that looks kind of knobby. And you've got some tyrosines here, and then these are kind of the available spots where you can phosphorylize these RTKs. So yeah. And then you'll notice that in this case it's tyrosine, but in general, the amino acids that are commonly phosphorylated, just in general, are serine, tyrosine, and threonine. So yeah, that's just something interesting to keep in mind. But yep. And then this is the thing that sets receptor tyrosine kinases apart from GPCRs the most. And that's that one complex, which is two of these receptor tyrosine kinases, can activate 10 plus transduction pathways and cellular responses. So this is really useful because means that one set can be really versatile, right? Whereas the GPCRs, just one type of receptor that leads to one type of pathway, and that's pretty much it. So you'll see that RTKs are involved in a lot of cancers. So for example, there is a cancer drug for, I believe, breast cancer and another type of cancer called Herceptin, and it's a protein that can bind to HER2, which is a type of receptor tyrosine kinase, to inhibit cellular growth. I believe the other type of cancer is liver cancer, but I could be wrong. But anyways, let's talk about how receptor tyrosine kinases work. So you start out with your individual monomers, and then your signaling molecule binds. And then what happens is these two monomers form a dimer. So they join together, right? This is what a dimer is. It's two monomers together. And what's going to happen is the receptor tyrosine kinases are going to phosphorylate each other's tyrosines. So this is pretty cool. So this one's going to phosphorylate this one, and it just kind of keeps on going. And this uses up 6 ATPs, right? It makes 6 ADPs, and that activates it. And what that allows this RTK to do is lead to a whole lot of cellular responses, because those phosphorylated tyrosines are going to activate various proteins and enzymes. So yeah, you'll see that these ones kind of seem more versatile, and then just like not that they're more useful, but they kind of do more at one time. And RTKs are, I believe, the most prevalent type of cell membrane receptor. All right, so the last type of receptor that we'll talk about are ion channel receptors. So the big type that we're going to talk about is the ligand-gated ion channel. This is a membrane receptor with a gate to a channel that opens and closes based on the shape of the receptor. So let's look at these pictures and the process. So you're going to start off with like the gate closed, and then whenever the signaling molecule binds, the gate's going to open because now our ligand-gated ion channel receptor changes shape. That lets things through, and that can lead to a cellular response. And then when the ligand stops being bound to our receptor, then the gate closes again, and then we return to the starting state. So yeah, you're going to see that this is really important in the nervous system because neurotransmitters are going to be those ligands that are going to bind to these ion channels at the synapse, and that's going to cause channels to open, and this is a great way to kind of cause responses within the cell. All right, so now let's talk about some intracellular receptors. So all of those receptors we just talked about, those are all on the surface of a protein, of a cell. They're in the membrane. And that's because those ligands are generally big and water soluble. So you'll see with our intracellular receptors that they're mostly located in the cytoplasm or the nucleus, and the signaling molecule involved has to be able to pass through the plasma membrane. So they're usually hydrophobic or small molecules. Hopefully that makes sense because those are the things that go through the membrane. So you'll see steroid and thyroid hormones, both of which are hydrophobic. And then you also see nitric acid pass, which is a gas. So basically anything that might be like a gas, something really small, something hydrophobic, that has a large chance of having an intracellular receptor. That is a popular question you're going to see on some tests where they're basically like, oh, which of these receptors is probably located within the cell, and then it might be like a receptor for some steroid hormone, and then that's going to be within the cell, like testosterone, which is this example I'm giving right here, secreted by the cells of the testes, and it travels through the blood and enters all sorts of cells. But what's interesting to note is that only cells with testosterone receptors actually respond. So you don't get like the same response to testosterone everywhere, and in some places you don't get a response at all because you don't have receptors for testosterone. So yeah, that's how it works. And then basically what testosterone does is it activates a receptor protein, and that receptor protein enters the nucleus to activate certain genes, and that's why testosterone has its effects. And in general, intracellular receptors, they might act as transcription factors, which are proteins that control the transcription of certain portions of DNA. So like that, these intracellular receptors can control kind of gene expression and what proteins are being made. All right, so now let's move on to the second step of signal transduction, which is transduction. So transduction, you're going to find, is usually multi-step, and this allows for more amplification and coordination, in this pathway. And usually what a pathway looks like is you've got a signal-activated receptor, and then, that activates a molecule, which activates another molecule, and you get this whole type of, like, cascade, and then you get these things called relay molecules, which are proteins that relay the signal from the receptor to response. So we would call all of these things relay molecules because they're kind of like the intermediaries between reception and the response. So yeah, and then kind of the big mechanism we're activating and deactivating by is going to be protein phosphorylation and dephosphorylation. So a protein kinase, like we mentioned, is an enzyme that transfers phosphates from ATP to proteins, and we're mostly going to see them interact with serine, threonine, and tyrosine, the amino acids, because those ones are the ones that are most commonly phosphorylated. And it's important to note that phosphorylating a protein may decrease activity, though we typically think about phosphorylation as like activation, right? And then But generally, yeah, that's how we're going to be activating our protein kinases. And a protein phosphatase, you see it labeled as this kind of purple little oval in this diagram. Protein phosphatases are going to dephosphorylate a protein, and that's going to inactivate it, so you can kind of start this cascade again. And protein phosphatases are really important points of regulation, because if you make sure that protein phosphatases are doing the work they should be, then you're not going to get a case where this pathway just keeps on going and keeps on going, and you're going to get way too much of a response. So yeah, pretty intricate cascade with lots of points of regulation. Alright, so let's talk about these things called second messengers, which are really important in transduction. And they're small, non-protein water-solubles and water-soluble molecules or ions, and they participate in both GPCR and RTK pathways. And we're going to talk about two examples, CAMP or cyclic AMP and calcium ions. So let's talk about cyclic AMP or CAMP. So let's talk about an example with epinephrine. So epinephrine or adrenaline is going to bind to a GPCR on the surface of a liver cell. And what that does is it activates adenylyl cyclase, which converts ATP to cyclic AMP. And cyclic AMP can increase like 20-fold in seconds. So obviously your cell is working very hard. So let's look right here at this picture. You see activated G protein goes and activates adenylyl cyclase, which makes a bunch of cyclic AMP. And cyclic AMP does a great job at broadcasting this signal. So it usually uses protein kinase A, which is a serine / threonine kinase, to kind of start off this entire cascade that leads to a cellular response. And then we're going to see that there's an enzyme called phosphodiesterase that converts cyclic AMP to AMP. So you can see that process right here. Adenylyl cyclase turns ATP to cyclic AMP, and then phosphodiesterase turns it into AMP. All right, so we're going to talk about how cyclic AMP is important for cholera. So cholera forms a biofilm on the small intestine lining, and that makes cholera toxin, and that modifies G proteins. And G proteins, they're going to be kind of rendered unable to hydrolyze GTP, and thus they always stay active. You're going to make a lot of CAMP. And what results from that is a lot of salts are going to go inside your intestines from the cells lining your intestines. That's going to draw a lot of water out via osmosis, and that's why cholera causes really bad diarrhea. So yeah. So now let's talk about calcium ions. So you see this used more commonly than cyclic AMP, although it does show up less on tests usually. It's seen in muscle cell contraction, secretion, cell division, and plant responses. So that's kind of a mem as well. Plant responses, this is a very common Science Bowl question where they ask for what ion is used in plant responses, and it's going to be calcium ions. So the kind of nice thing about calcium ions that makes them really ideal for this purpose is that you have like basically no calcium ions within the cytosol, but you've got a lot of them outside the cell. So it does not take a lot of calcium ions to go into the cell for your cell to detect a change. So, and this is maintained by actively pumping calcium ions into the ER or out of the cell. And yeah. So basically what happens is you got this molecule called PIP2 in your membrane. It is a type of phospholipid, and you've got a GPCR that's activated that activates phospholipase C, which cleaves PIP2 into IP3, which is our second messenger. That's, I guess, acting first in this pathway. And then you are left with this thing called DAG, and that goes and acts as a second messenger in other pathways. And then IP3 basically goes and activates a gated calcium channel, which is one of those ligand-gated ion channels. And then calcium rushes in, and that's going to activate a bunch of proteins, lead to various cell responses. So we actually see 3 second messengers in this pathway: DAG, IP3, and calcium. So yeah, that's pretty interesting, but calcium is definitely kind of a very talked about second messenger. All right, so now let's talk about the response that is caused by all of that cellular transduction. So one thing we can do is regulate protein synthesis. So this is going to happen in the nucleus, right? And we can also regulate protein activity, and that's stuff that happens in the cytoplasm. So this can be opening or closing an ion channel, and be a change in metabolism. Like with epinephrine, the goal of epinephrine in a liver cell is to tell your liver to break down glycogen, and that's going to release a lot of glucose in your blood, which you really need if, you got that fight or flight response. You're going to need a lot of glucose to support all that activity, right? So if you look at this picture, it just shows kind of like how you can have a growth factor, and that ultimately leads to activating a transcription factor to transcribe a gene, and that's going to lead to that gene being expressed as a protein. So yeah. And here's just some more on a response. So we're going to see this thing called signal amplification, which is what it sounds like. So we get signal amplification because proteins are often active for long enough to process multiple substrates before becoming inactive. So let's just say one protein is able to activate 5 proteins while it's still active. So now we've increased everything 5-fold. And we also see that pathways can interact with each other. So if you look at this picture, we see that this is kind of our standard case where one pathway leads to one response, but our pathway can split. We can have multiple pathways kind of inhibit or support each other, and we can also have the same pathway with a different signaling molecule, and that leads to a different response, or the same molecule with a different pathway leading to a different response. But yeah, those are all things that can happen. And this picture here is supposed to show signal amplification in the epinephrine case. And basically, we see a lot of amplification. I would recommend that you have like a decent idea of where you're seeing amplification in this process, particularly adenylyl cyclase, basically that gets amplified by cyclic AMP like about 100 times, right? But that means one epinephrine molecule causes how much is that? 10 to the 8th glucose molecules to be released in the blood. And that's pretty crazy. So yeah. And then lastly, scaffolding proteins can carry relay proteins together, which enhances speed and accuracy. So we see our scaffolding protein right here. It's carrying 3 different protein kinases. And instead of just having all of these parts like float around in the cytosol, waiting to kind of activate each other, if you're holding them together with your scaffolding protein, it just goes like boom, 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 very fast, very efficient, and that makes the whole thing kind of just easier. So yeah. And then this is going to be the last thing we talk about, apoptosis. So this is programmed cell death, and might not seem like it has much to do with signal transduction, but it really does, because it brings together a lot of these concepts, and it's also just a very central process in cells, right? So basically, you see apoptosis in infected, damaged, or old cells. And what happens is you're going to chop up all your DNA, chop up all your organelles, and then you're going to see blebbing. So in this picture, you see kind of blebbing right here, but it's basically where your cell shrinks and you get lobes. So I feel like blebbing is one of those words where the word really describes what the whole thing looks like. Because bleb makes me think of blobs. Basically, it's like your cell turning into little blobs. So yeah. And you're going to have your cell parts put in vesicles, and that's going to be digested by scavenging cells. So pretty much it's a planned death that makes it so, you can recycle a lot of the parts, and so the cell's contents don't end up like harming other cells around it. So let's look at apoptosis in C. elegans, which is a nematode, which is a roundworm. And it's a really, really commonly used animal. in kind of lab experiments because it's only got about 1,000 cells. Scientists have sequenced its whole genome. They're very small, easy to breed. So they're pretty much an ideal lab animal. And what's crazy is scientists figured out that apoptosis occurs exactly 131 times in a C. elegans that's developing normally. And we see the process of apoptosis is led by the CED3 and CED4 proteins, which come from the CED3 and CED4 genes. And you've got this CED9 protein on the mitochondria that inhibits CED4. So here's kind of like the inactive form when the cell is like doing all right, no apoptosis signal. You got your CED3 and CED4 that are inactive, and then you've got your CED9, which is active, and it is preventing CED4 from becoming active. All right, so once you get that apoptosis signal in our C. elegans, what's going to happen is the signal transduction is going to lead to CED9 becoming inactive. And now CED4 can be active. It stimulates CED3 to be active as well. And then you're going to lead to the activation of lots of proteases, which break down proteins, and nucleases, which break down nucleic acids. And then kind of like a mem is caspase. Caspases are the main protease. And how I like to remember caspases having a role in apoptosis is like Casper the Friendly Ghost. Casper, ghosts are dead, right? So like caspases, they have to do a cell death. So yeah. And then this is how it basically goes in C. elegans, and it's not too different in humans and other animals. So you're just going to see more caspases. There's 15 different caspases. There's multiple pathways. So it's just kind of like a more complex version of this. And something kind of important to note is that mitochondrial proteins can make pores in the outer mitochondrial membrane during apoptosis, and this releases more apoptosis promoting proteins, including cytochrome C. Now, if you remember from cellular respiration, cytochrome C is one of the electron carriers in the electron transport chain. So that's what it normally does, but in apoptosis, where the mitochondria are going to, kind of break apart, cytochrome C promote apoptosis. And lastly, something to note about apoptosis is that the signal doesn't have to come from outside of the cell, it can come from within the cell. So, for example, the nucleus can send a signal to the cell that the DNA suffered really bad damage and there's a chance that this cell might become a cancer cell if it's allowed to reproduce, and thus the cell will decide to commit apoptosis and shut everything down. Or the ER can send a signal that there's been really bad protein misfolding, which can lead to a lot of issues like prion diseases that comes from, bad protein misfolding. So those are all things we want to prevent. So apoptosis gives us a chance to basically let that cell die before it can influence other cells. So yeah, that's a very common function of apoptosis. And let's see. Apoptosis, just some other application, is that they're important for the development of hands and feet and paws in animals. And what happens is when you're developing as an embryo, your hand looks kind of like an Animal Crossing villager's hand. It's like kind of circular round. It's like you don't really have fingers. And the way you get fingers is the cells in between your fingers die. And that's where your apoptosis is coming in, because those cell deaths are planned. And what you're going to see is that there's less apoptosis in organisms with webbed feet, because those webs are like, kind of the cells that die in organisms with like hands like ours, right? And apoptosis is also involved in Parkinson's disease and Alzheimer's disease, kind of faulty apoptosis, right? And then faulty apoptosis can also cause cancer because, like we said, the nucleus can send a signal to the cell that there's been really bad, DNA damage, and that the cell should commit apoptosis. But if that doesn't happen, that DNA damage can lead to changes that would lead to the creation of cancer cells. So yeah. All right, that is the end of the lesson. Thank y'all guys so much for watching. I really hope this helped. And Please ask any questions, and if you feel like this video helped you, please leave a like and a comment and subscribe for more content. Thank you.